conferencia de la ciencia más allá del aula. Hoy tenemos la visita desde Singapur del doctor Alvin Heah. Él es vicepresidente de ventas de ESCO Life Science Group. La conferencia será en inglés, pero les voy a platicar un poquito de la trayectoria de Alvin. Él se incorporó a ESCO en 2021, que es una empresa de ciencias biológicas líder en la fabricación de equipos de laboratorio y biofarmacéuticos, también de dispositivos médicos de fertilización in vitro. Desde entonces ha ocupado varios puestos en ESCO, entre ellos el de ingeniero de exportaciones, director de desarrollo empresarial, director de operaciones europeas y director de proyectos y desarrollo empresarial. Hoy es el actual vicepresidente de ventas de ESCO Life Science Group, que tiene presencia en más de 100 países. Él es responsable de las ventas a nivel mundial, excepto China, lidera proyectos para ESCO Scientific y ESCO Pharma, una subdivisión de ESCO Healthcare. Durante su tiempo gestionando este desarrollo empresarial en Europa, se centró en establecer la presencia de ESCO en la región, comenzando en los Países Bajos y luego en el Reino Unido. Aparte de Europa, también ha gestionado distribuidores en el noroeste de Asia, en el sureste asiático y en América Latina. Alvin también es un certificador de campo de gabinetes de bioseguridad acreditado por la NSF. Obtuvo su grado de ingeniero en eléctrica y microelectrónica en la Universidad Nacional de Singapur y recibió la beca Asian PU Scholarship. Alvin nos platicará un poco sobre el alcance de ESCO y sus preocupaciones para el desarrollo de la industria farmacéutica y pues bueno, le estamos muy agradecidos. Gracias, Alvin.
Thank you very much. It's okay? All right. So I'm very happy to be here. I have about an hour or so with you. And let me start by introducing what I would like to achieve today. To basically share some of the solutions that are used in GMP-related application or field. We will change between the concepts of biosafety and containment, in particular isolation containment, as we progress through these slides. So I wanted to just begin by showing you a slide on a device called pharmaceutical isolators. They are used in, as you can see the name, pharmaceutical manufacturing companies in many different kinds of applications. The pharmaceutical isolators must comply with GMP guidelines. Pharmaceutical isolators are mainly divided into two types. One is for containment, mainly to contain very toxic substances. And the other one would be aseptic. That means you have to manufacture drugs under very sterile, the word aseptic means that, conditions. And the construction of this equipment, if you want to follow GMP guidelines, are listed down here, right? They must be of a certain thickness of stainless steel. They must be very smooth in the finishing for easy cleaning, just to give you an example. And one of the topics about cleaning will have to involve hydrogen peroxide decontamination. So again, I have no way to interact with you, right? But it is not a simple wiping with alcohol the surfaces to clean. So you have got to use hydrogen peroxide as an example to achieve your 6-log reduction in your potential contamination. So that is actually something that I would like to leave with you as an example. We have a lot of strict regulations. I do know that everyone knows the US FDA. I hear there is a very powerful organization. So if you are listening to me, I know you, Kofi Priest. I know you. I know who you are. Okay, but there are actually a lot of approvals that have got to go into the building of such an equipment. A lot of the materials that we use to make an isolator for pharma needs to be FDA approved. And there will be the concept of following procedures step by step to qualify and validate the equipment. So we come across, documents like IQ or OQ. This is actually just an example. Okay. Now, when we talk about qualification of the equipment, we have to understand that this equipment normally gets produced at a factory. And most of our customers, when they purchase this equipment, would insist to inspect the performance of the equipment at the factory. They will travel all the way, thousands of miles by aeroplane, to spend a week with the manufacturer and check the requirements against the performance against the requirements one by one. And this step is called a factory acceptance test, or an FAT. So this photo here shows you a filling line isolator. So when we had COVID and we had this rush of vaccine producer, the Pfizer, the Moderna, and all of these are in vials, they actually get filled aseptically in isolators like these. And they actually are simulated at the factory with pseudo HVAC. You can see here a duct that is connected to a temporary HVAC to simulate the actual working performance of the isolator. Yeah. The client will come and inspect, spend a few days, right? And there will be lots of arguments sometimes. Saying that this shouldn't be like that, this should be repaired, rectified. So it's actually a very thorough process. Okay, now this photo here shows you the back part of an isolator. We covered up with a blank piece. I thought I'd just show you this because this here is a future connection to a device called a lyophilizer. So a lyophilizer is a device that freeze dries drugs at the end. So some drugs are actually freeze dried, and that actually is basically how you would connect a freeze dryer, a lyophilizer, to an isolator. In the process of making sure the user is comfortable working in an isolator, it is almost impossible to cater for small, medium, large. I am large. Some ladies are medium or small. So there is actually what we call an ergonomic trial, because you can have the best isolation containment machine, but the ergonomic trial is the one that will make sure the dimensions, the reach, the height, the space are all optimized before you set up the equipment in the factory. Yeah. So again, this may not be relevant to your research or your studies. These are all tools, engineering tools that you find in industry. So I hope that I can enlighten you with the process. This process is called an ergonomic trial. We use cardboards, we use boxes, we put them up together like Lego, and we really invite the customer to come down, to reach into it, to work with it. Yeah. I hope my English is okay for your understanding. Yeah. Okay. Another topic I would like to share with you is also, and coffee please. Hello, I'm here. It is the very spectacular, very sexy, exciting development in cell therapy. Cell therapy. So cell therapy is a kind of therapeutics. It's very novel, right? You basically use materials like your own stem cells. But basically, these cells need to be cultured. And if for human use, GMP guidelines should apply. When we talk about cell culture, mammalian cell culture, we are talking about, handling it in an aseptic environment. Normally, you would imagine a biological safety cabinet. You would imagine a centrifuge, okay, that would do your centrifugation. You would have your CO2 incubators to incubate the cells. You might have a fridge or a freezer to store the samples, so on and so forth. So isolators are actually now used to combine all the processes to give you what we call a cell processing solution, right? So everything here is done in a closed aseptic manner. The CO2 incubator is clean and decontaminated with, not with alcohol, with hydrogen peroxide. Yes? The centrifuge inside, you cannot see this. I will show you some photos later on, is also very airtight, very resistant to hydrogen peroxide. And so this is yet another example I would like to share with you. All of these machines, when combined together, must comply to the same set of GMP rules that pharmaceutical companies go through. Isolators can be configured. It can be configured, according to the size of the space available. There is no end to the imagination. Yeah, you can connect anything, right? But you have to test it. It must isolate. It must be tight. So my point here is that people think about isolators as a working bench. Please, it does not work like that in industry. They can come in many kinds of configurations: L, W, a straight line, so on and so forth. Okay. Okay. In all of from what I've seen over the years, when people think about isolators, they think of a box with some filters inside and some glove ports, correct? There are many different kinds of configurations that we can have in an isolator, but the principle, which I hope to share with you later on in the next section, will apply. The other exciting area that uses isolation technology is in the area of radiopharmacy. Nuclear medicine, for another substitute word. So, radiation therapy using radioisotopes is proving to be very popular. It has shown very good results. Let me give you an example. The radioisotope lutetium-137, I think, please don't come up. I think it's lutetium-137, is actually used now to fight, and hopefully cure prostate cancer. Yeah. So these radioisotopes is a radiopharmaceutical. It is actually produced in isolators, like what you would find in pharma, traditional pharma, vaccine companies. The difference is that radiopharmaceuticals emit radiation, and there is a big safety risk for the user. So we have got to imagine that the walls of the isolator are protected with lead. I know this word in Spanish, plomo, yeah, lead. So lead actually is inside every surface around the isolator. So that is the main difference. I want to give you an example. One typical isolator without lead is about 2 tons, 2,000 kilograms. Isolator like this would be 5 or 6 times the weight. So when we talk about the preparation of radiopharmaceuticals, we like to believe that everyone, including the authorities, would respect that these need to be produced in accordance to the same GMP guidelines. This actually is something that ESCO started the isolation technology with. It is close to my heart, right? This is actually a biological safety cabinet joined up with a class 3 biological safety cabinet / isolator. This configuration here is used mainly in public health laboratories, Center for Disease Control. This isolator here is highly negative. It is like, like if you shoot the glass, right, air and there's a hole, air will not come out. It's highly negative. It is used a lot during the times of COVID. Before that, there was SARS, there was bird flu. So this actually is another shape and form of isolators. We work a lot with the people at the Ministry of Health. Now, back to the traditional pharmaceutical companies. This actually is a very normal configuration, but it's not normal in, I believe, any one of your labs. When you do your research or studies, you won't see this. This actually is a configuration which combines the final isolator connected with a fume hood, a chemical fume hood. So you would prepare your chemical, your solvents, your formulation, and then when you actually want to do processing of the API means active pharmaceutical ingredient inside the isolator, you can do so in a very safe way. Safe. Okay. I chose this photo here to show you an accessory. It's called a BIBO filter. BIBO, yeah? BIBO is a bag in, bag out. It is a filter, it's a HEPA filter that traps mainly toxic powders that cannot be deactivated by any gas. You have a bag around it. So these filters need to be changed. And when they do get changed, we don't want the engineer, the poor engineer, to inhale whatever toxic materials from that filter. So there'll be a bag for it, right? So this is actually a live photo. I cannot quote the customer, sorry, it's confidential, in a pharmaceutical factory. So it looks like this, right? It looks like this. The responsible user who wants to protect the safety of himself and the fellow workers will be creative enough to engage in companies like us to think about the process and make sure that at the end of the day, everyone goes home safe. Everyone goes home safe. Okay. With regards to this photo here. This photo here is a biological safety cabinet. It is an open system. It is not an isolator. Biological safety cabinets is not an isolator. It is more at risk of containment failure. It does contain, but not as strong as the isolator. But the biological safety cabinet is a very loyal safety equipment you can possibly ask for. Very loyal, very functional. Okay. Biological safety cabinets can also be modified. This was actually the initiative of the government of Singapore during pandemic. The PCR testing was too slow by human, too slow. So what they had to do was to find a solution to integrate a robotic system into an enclosure we call a biological safety cabinet. There is a link here. It came out on television, and we are very proud. We are very proud to actually bring biosafety and help, the more rapid PCR testing during the pandemic. Biosafety is the concern of not only WHO, but many other United Nations organizations. And I wanted to share with you that if you are in the field of biosafety and you are passionate about what happens in a lab, how can you prevent from getting the infections yourself? Well, we are very fortunate to have organizations like these. And this one here, I'm particularly very emotional. UNICEF also contributes to ensuring that all developing countries, particularly I must mention Africa, all the hospital, the healthcare setups are properly equipped with, number one, biosafety cabinets. These are actually awards, contracts that we get long-term, 1 year, 2 years from these organizations, and we actually deliver these to very remote places. Some countries, the name I cannot even spell. Whatever it is, right? Every product that is actually linked with safety must be regulated. So for biosafety, the American standards is very strong. NSF is really influential in the area of biosafety. NSF has recognized that the equipment must be tested once a year. But who tests the equipment is also very important. If you bring an unqualified engineer to test the equipment, then you would have a bad result and the machine will not function in a good way. So NSF has got programs to train up engineers. Now I know many students in a similar position like you in other countries who actually gain an interest and sign up for these programs to be tested, to be qualified as a biosafety testing engineer. That is actually something I would like to share with you. Okay, but let's come back to the biological safety cabinet. In the pharmaceutical setup, it is a grade A sterile environment, but it's open, there's no gloves. This is a typical setup in a lab, how a biological safety cabinet looks like. Some of you may be working in such a lab. But let me tell you that there are customers who actually read about the GMP guideline. Notice when I showed you in the very first slide, the GMP guideline promotes the selection of good stainless steel material. Stainless steel 316, 4 millimeters. And here you have a photo of a biological safety cabinet. It's not really an isolator, but the customer actually invests in upgrading the material. So they cannot fully follow the guidelines totally, but they have an attempt to customize this. This is just a little demo to show you that if somebody uses a microscope inside a biological safety cabinet, just by putting the microscope inside, I can bet you, if you use some kind of device to check for containment, the containment is very bad. There is actually a way to integrate microscopes into biological safety cabinets without compromising the containment. Now, coming back to process development, or R&D in pharma, it is very common to want to see that your molecule or your formula actually sees the light of day as a pharmaceutical product. So we always hear about aseptic fill and finish. So you got to fill your drug product into vials, syringes, and during such a phase, you can't possibly afford to buy expensive pharmaceutical isolators, too expensive. So a smaller version is what we call a biological safety cabinet that is floor standing. We call it the bio booth. I do not know how many of you have seen a biological safety cabinet. Many of you have in a lab. But to see a biological safety cabinet that looks like this, with a door that opens up, full length, it's very uncommon. But it is a biological safety cabinet, and you put inside filling machines, small filling machines inside to do your process development. That is actually something I like to share with you. The UV lamp can be turned on as per normal, as per your biological safety cabinet. Okay, I would like to share with you the importance of GMP guidelines, the influence it has on people. So this is actually a photograph of a typical biological safety cabinet, but if you look closer, there's actually a particle counter probe. A particle counter probe. So particle counter counts particles. It checks for sterility. You don't find particle counters mounted inside a biological safety cabinet in a lab. You don't. You don't. But in pharma, this is very, very common, I must say. So you have here the inclusion of a particle counter. We call this a non-viable counter. This is actually a microbial air sampler, right? That actually, checks for presence of microbials, and these are all integrated in real time. Do you find this in aseptic isolators? Every time. Every time that you have a pharmaceutical isolator, every work chamber has got a pair of these. Do you find these in biological safety cabinets in a lab? No. But the trend is moving towards equipping biological safety cabinets with these two devices. Okay, biological safety cabinets in a GMP facility can look really, really different from those that are inside your lab. Skip here. Yeah, safety cabinets can equip big devices, so they can be customized. Handling of toxic powder in drums. So for oral solid OSDs, tablets, they all come from bulk powder, bulk, B-U-L-K, bulk. Bulk powder normally is stored in drums, right? You have to open up the drum cover, you have to, tear open that plastic and do your scooping, do your weighing, whatever you want to do. And there is actually a risk. When you do this, powder will tend to, fly upwards, be attacking your face, your respiratory pathway. So this is a very, very easy to understand example. It is a drum. You put it on a trolley. This trolley can move upwards into a safety enclosure so that you can have good containment. That is actually an example. So you have here live pictures. I can't tell you the pharma company is a very big one. You have radio pharma. Okay, in pharma, sometimes in the processing of your drugs, solvents are sometimes used in large quantities. Solvents can be flammable, that's one. Sometimes you use powder in large quantity. Powder, when you expose powder to light, it can explode. So there would be a need to recognize that some areas in pharma manufacturing will be designated ATEX zone. ATEX is explosion proof. So when you have this kind of a zone, every containment equipment inside, whether it's an isolator, a fume hood, a biological safety cabinet, everything electrical inside must be ATEX rated. ATEX, A-T-E-X. Okay, so I can't elaborate a lot. Time is short. Do the research separately. Okay, there's fire extinguishing systems, right? This client here was working with some kind of a spray, and this spray is actually flammable, and so we must put in fire extinguishing system on the fume hoods. Last but not least, the pharmacies. Now, say a patient who has cancer goes for chemotherapy. Chemotherapy, he or she will have a customized treatment. So, there will be a compounding. So there will be a drug A, B, C, whatever components, and then you will just mix them all up, compounding, and they are performed, in places, not in the pharmaceutical factory. No, they are done in hospital laboratories. Some of them are commercial pharmacies. This is actually an example of the kind of equipment that is being used to compound these drugs. So the question we have to ask ourselves is, this is the final step before it gets delivered to the patient. Must this equipment be following GMP? Yes or no? Yes or no? Right? Must it be prepared in a clean room environment? Yes or no? Now, I would not proceed further. Being very passionate about safety, I have seen many hospitals doing pharmacy compounding in a very bad situation. Bad. No clean rooms, no proper gowning, mix here, mix there, right? Okay, of course, all of these products are. You said something? No. Oh, I'm sorry. Yeah. Okay, so these actually are a bit unfortunate, but I can assure you that full GMP guidelines in a pharmacy, not happening. Okay, now this here looks like an isolator. It's very popular. It's called a RABS. RABS, R-A-B-S. RABS is an acronym for Restricted Access Barrier System. It is not an isolator. So if you do a pressure test on these products, the RABS, there will be some leaks. It contains, yes, by inducing pressure inside the RABS, whether it's negative pressure or positive pressure with respect to the room. Right, okay. So nothing wrong with that. Yeah, it's just a little bit sad that there is not enough guidelines or audits or checks. I know how the likes of FDA, coffee priests, they make life so difficult for the pharma customers in industry. Sorry for that, but I think there is a little bit relaxation, in the hospitals. And that's sad. That's sad, right? So I do not know how many of you here are probably going to branch out into the area of pharmacy, right? But this is something to take note of, that engineering safety does apply also to people in the pharmacy. You mean the pharmaceutical engineer's life is more valuable than the pharmacist in the hospital? I disagree. Yeah, we are all, equally, important to be protected. That actually is that last part about sharing with you practical solutions. I've shown you a mixture of isolators and open systems, mainly focusing on biosafety. Okay, so I'll take a pause here. Can I take a pause? I can't take questions, right? I can? Okay. Any questions, and I'm sorry I sound like that because I have had little sleep, jet lagged. Apologies, disculpe-me, lo siento. Yes. Are there any questions? What is the time now? Just a moment. Slide.
¿Alguna duda, alguna pregunta, algún comentario?
¿Más tiempo?
Sí, porque empezamos también un poco tarde. Sí, unos 10 minutos. Empezamos tarde.
Yes, so in the interest of time, I would like to share a few more words and photos and pictures to show you what actually is important with regards to isolators. So let me just give you an example. I don't specifically explain this purely from a cell therapy point of view, but isolation in general must conform to the same few guidelines. Okay, so basically, in an isolator, in the insides, you have to maintain ISO class 5 or grade A air cleanliness. In order to do that, you must have full HEPA filter coverage inside the work zone. The laminar flow is mandatory for aseptic isolators, and you have the possibility to configure the isolator to operate between positive or negative pressure depending on the toxicity of the product. If it's very dangerous or hazardous, you switch to negative pressure regime. And with regards to the airflow, you can actually operate the isolator in a recirculating manner or a total exhaust manner. Total exhaust means that the air, once filtered and passes through the product, will never enter the work zone again. It will go out immediately, and this actually is expensive. Recycling the air is a little bit more cost effective, but recycling of the air within the chamber has risks, especially if the products that you work with cannot be totally filtered by the HEPA. Okay, so I will just go through here very quickly. Okay, let's come to this point here, right? Every isolator, before you can start work, needs to be verified to be leak tight. So there is a test that has to go into this. It's called the pressure leak test. The pressure leak test is a process to introduce air into the chamber in very high pressure. And we have to hold the pressure and see if there's no decay in the pressure. So as long as the decay pressure test passes, you can proceed to the next step. Okay. Then we talk about regulation. Every step you take and work with the isolator must be recorded. So there is this standards that we have to follow. It's called the 21 CFR Part 11. And this actually is a requirement that governs what kind of controller you select. I'm not promoting Siemens here, but Siemens is a German brand and it's actually very famous in this area for producing very good quality HMI, human machine interface, which is actually driven by a PLC. PLC is programmable logic controller. So you have here the ability to have a touch screen in front of you, and you can monitor all of the parameters. You can get reports for all of the logs and the alarms and the events that happens within the isolator. So in summary, we actually have this audit trail here to be very commonly requested for any isolator that comes into the factory. There's also different security access levels. So you have the user, the supervisor, the admin. Okay, there is actually all built-in features that a HMI PLC can provide. We also have the another, what we call a sign-off, an e-signature, right? Where, it is possible with the 21 CFR Part 11 compliant control system and the software. This is actually going to be what I learned recently. All of these reports are preferred to be in PDF, non-editable reports. Reports are very important for the validation team. How you download the report is the key. Today, many pharma customers disallow the use of the USB drive, put it inside the isolator, download the reports. That is kind of like, not very secure. Even though you put security access to who can download, it's not very secure. So we actually see now the isolators being connected to building management systems via a LAN cable, right? Gloves need to be tested. Gloves, they are actually pressurized with compressed air again, using standalone machines, and they are held for about 5 minutes each glove. So you would do your glove testing 5 minutes for each glove before you are allowed to start working. Okay. Yeah, so what if you have 24 gloves? You are going to, it is very long cable, so there are modern gloves like this one here called the wireless glove testers. All right, I'll just skip here. And just to share with you here, the biodecon system. So the biodecon system uses H2O2. And H2O2 actually, it comes in a bottle like this. Isolators come with a compartment that houses this bottle, and it will actually be sitting on a weighing scale. And this is a 500 ml bottle. So just to give you an example, to guess, H2O2 for a 4-glove isolator, it will take about 1.5 hours. It's that long. And it will use up to about 40 milliliters, 40 to 50. So one bottle will allow you to gas about 10 times. And you have to gas your isolators before you run your batch. We have to actually verify that the cycle is good. So we paste indicators, biological indicators, all over the isolator work area to make sure that there's no growth. This process takes about 3 to 5 days to get the results. Okay, that's it. And of course, you have in every chamber a pair of these probes, right? Probes. Okay. This is how a particle counter looks like, right? And I think these are just some photos. In quality control, there is this test called in microbiology, it's called the sterility testing, right? And they use isolators as well. Some people call it the bioburden test, but basically you do have this function in pharma. Okay, thank you for my 10 minutes. I'm very happy to have been able to say some things to you today. Thank you for attending. I hope this information is somewhat, meaningful, and hopefully it can spur your interest and imagination to find out more about engineering, safety, and containment. So thank you very much. I wish you all a very good day. I hope to see you again soon. Thank you very much. Bye.
Thank you, Abel, if there's no more questions, okay, there's one question over there.
One question. What do you think is the country or the countries with the more strongest rules in biosafety, or with more or with the best installations about biosafety in the world?
Wow, I can be getting into trouble. I've seen many installations, and I feel that the Europeans, the Germans, the Swiss, to answer your question directly, I feel that they have the highest standards and adherence to biosafety. This is my personal opinion. And with regards to Estados Unidos, maybe not as good, although the theory is that they are very good, but compared to the Western Europeans, it's, I believe, a very big gap. I mean, is that...
¿Alguien más?
Hello, Alvin. Nice to meet you. And in your opinion or in your experience, what is the situation in Mexico about the biosafety?
In the area of biosafety, I have a few observations, and please don't kill me. Yeah? Don't come after me. First, I believe that the engineers that are allowed to maintain the equipment are not qualified. That's number one. And number two, I believe that the users are actually not working in synchronization with a health and safety office of a facility to, number one, primary reason, make sure that the cabinet is maintained at least once a year. Like the car, you know, maybe most of you drive the car, you have to change your engine oil, you have to do some maintenance, maybe 6 months or 1 year once. There is no difference with the biosafety cabinet. Let me tell you that the containment goes down. Every day you use it. Every day, the level of containment goes, it's not immediately abrupt, but it goes down. So that is actually a couple of observations that I have. But on the positive note, I have been to Mexico many times. I've met many users. I feel that the awareness, once there, people are serious. People are mature and adult about it. Yeah. So I'm very hopeful that things will improve in a organic way, hopefully quicker. That's my comment on that. Yeah. Yeah.
¿Alguien más?
So please join me to thank Abel for this talk and this concern about biosecurity that we learned a lot today. So I will give you this recognition for your talk. Thank you. And please a round of applause for Abel.